Hello students. In our last class, we have discussed about the introduction of cell as the fundamental unit of life. Today, we will discuss about a very important component of the cell, which is cell membrane. Cell membrane. This cell membrane is also known as plasma membrane, plasma membrane or biomembrane or plasma lemma This is the structure which create the boundary so and isolate the cellular system from the external environment and from the adjacent cell also. That is, it isolates the cellular system so that all the cell organelles along with plasma membrane can perform the life processes efficiently, and this can sustain. So it is very important function that first importance of cell membrane is that it isolates the system. Then the second thing, this cell membrane is a porous structure. Okay, and a very important thing is that it is selectively permeable. Structure. What is the meaning of selective permeable? Selective means it selects which material or which substance is good for cell and which is harmful for the cell. So it is selective. Then permeable, any membrane which allow the movement of substance from or through it. Okay, if it is allowing the substance, mean it is permeable. Okay, allow the substance to move through it, but it should be selective. Because of this nature, this plasma membrane play very important role in regulating the transport of substance toward the cell and from cell to external environment. Because it is regulating the transport of substance, so due to the selective permeable membrane, we can just write the second function of it, regulate transport of substance. Okay, so let's see what exactly or how the transport of substance takes place. For this, we have discussed about the term diffusion in our previous chapter, which was the metacellular surrounding. What was a diffusion? Diffusion. It is free movement of any molecule molecule from higher concentration, from its higher concentration to lower concentration. Let's see one example. Any unicellular organism gets oxygen from the external environment, or in our body the cell gets oxygen from the capillaries and give the carbon dioxide to that capillaries. In case of the unicellular organism, the carbon dioxide is given to the external environment and the oxygen is taken in. How is it possible? Suppose it is external environment. It is cells, cellular, and it is cellular system, and it is cell membrane, this boundary. Okay, cell membrane. What happens in external environment? Concentration of oxygen is high. Due to respiration, concentration of carbon dioxide is high inside the cell. So, as per the diffusion, what will happen? The oxygen will move in from higher concentration to the lower concentration through this permeable membrane, which is allowing the oxygen to come in, and the carbon dioxide will move in external environment or move out. So, due to this diffusion, gaseous exchange, which is very important for the respiration of any cell, takes place. So it was one importance. Then the nutrition can also be absorbed because of this diffusion process. Okay, plant roots or plant cells and roots, or I should say plants will absorb water by roots by this process. Okay, so we can just observe that diffusion is very important, which is going to be regulated by our cell membrane. Now there is the second case, which is a special type of diffusion, that is, so let's see some special type of diffusion. What is this? When this diffusion is taken place by the water molecule, just see this water molecule. Okay, first point, diffusion by water molecule from higher water concentration to the lower water concentration, through semi-permeable membrane. Okay, these three conditions are mandatory. Then it is known as osmosis. Okay, osmosis. This osmosis is very important in case of living cell because it is a characteristic of living cell. Just see how it affects some activities of cell. Before this, I would like to explain the term water concentration. Suppose you are taking two beakers, A and B, having same amount of water. In beaker A, you are just putting 1 tablespoon of sugar. Okay. In the B, 2 tablespoons of sugar. Although the amount of water is same in both the beaker, but the concentration of water is higher in A than the B. That is, we can, in short, or just to get idea, we can say that the dilute solution of water is having more water concentration. Okay, it is dilute, but it is having the more water concentration. Okay, so let's see how the cell react in different concentrations of water. So, by following this osmosis process. In first situation, there are mainly three situations. In first situation, we are putting a cell, it is your cell, and a beaker having the concentration of water, more than cell, means here the water concentration is less. Okay, water concentration is less and here the water concentration is more. What will happen? The water will move out. Okay, having the water concentration is less, here you are getting, and here the water concentration is high. So water will move from cell to the external environment. The cell having, or the anything, if any solution, an external solution having the concentration more than the cell is known as hypertonic solution. This solution is known as hypertonic solution. I said concentration of this external solution is more. Concentration, not the water concentration. If concentration is more, then water concentration is automatically low. So, if the concentration of external environment is high, then that solution is known as hypertonic. That is, in short, we can learn hyper or high to cell. What? Concentration. If the concentration is high comparative to cell, then that solution is known as hypertonic solution. In this situation, the water will move to the out, and this process is known as exoosmosis. If this cell is animal cell, then what will happen? The water will move out and the animal cell will shrink. Okay, it will shrink. So it was the first situation. About plant cell we'll discuss later. Mainly here it is animal cell due to exosmosis. Now in the second, so first it was hypertonic solution. Now come to the hypertonic. Second situation, which is if your cell is kept in a solution having same concentration to the external environment. Here the concentration is same. That solution is known as isotonic. Iso means same. So same to the cell. Then the concentration is said as isotonic. What will happen? There will be no net movement of water. Although there will be movement of water, but in both directions, but the net movement is zero. Means the equal amount of water will go out and the same amount of water will come inside the cell. So the shape of shape of cell remain unchanged. Okay, so it was about this isotonic solution. Next and very important thing, come to the if a cell is kept in a solution which is of less concentration. Okay, less concentration. I am saying that less concentration means high water concentration. Water concentration. Less concentration means hypo. Hypo means less. Less to the cell. Okay, hypo to cell. So, what will happen? Water will move inside. Okay. This type of movement when the water comes inside the cell is known as endoosmosis. Endoosmosis. Due to this endoosmosis, as the water is coming in, what will happen? The cell will swell. Animal cell, if it is animal cell, it will swell. If there is more than this condition, if more water is coming in, then after some time it may burst. Okay, so it was the case of hypertonic solution. Clear? So it was about the how it regulate the movement of, how the cell membrane regulate the movement of materials. Now the third very important thing about the cell membrane is that its flexibility. Flexibility. This structure is very flexible. Due to its flexibility, I am giving you an example of amoeba. This something like this structure. These are the pseudopodia. These are nothing pseudopodia. Pseudo, I have told you earlier, pseudopodia means the false feet. It can appear and disappear depending upon the situation. What are these? These are nothing. These are the cell membranes. Cell membranes projection. Outward projection it is. Why is it possible? Because it is very flexible in nature, and due to its flexibility, the amoeba can engulf the food inside. Means it can do for phagocytosis. It can eject out, if means excretion can be done, or we call it exocytosis can happen just because of this. So this flexibility of cell is very important. Same in case of the WBC, shape is changing, polymorphic it is just because of this plasma membrane. Okay. So it was about the plasma membrane. In case of plant cell, it was the most important thing which we should just know. Then after this, one more covering we find in case of plant cell. What are these? It was the cell membrane, cell wall. Cell wall is what? It is basically present in plant cell. Plant cell made up of complex form of cellulose. Cellulose complex form. Okay. What does it do? It provides the protection, support, and one more thing very important, it can withstand the change in osmotic pressure. Osmotic pressure. Because it is rigid structure. Rigid structure. Okay. If we keep the plant cell in hypotonic solution. Hypotonic solution. What will happen? Water will come in. Cell try to swell, but due to that cell wall, the wall pressure will created on the cell membrane, so the shape will remain unchanged. Shape unchanged. If we kept it in hypertonic solution, exoosmosis, osmosis. In case of animal cell, it was shrinking, but in case of plant cell, it is cell wall. Due to the exoosmosis, the plasma membrane will leave the corners of the cell wall but remain attached at some corners. This particular phenomena is known as plasmolysis. It may be temporary if we provide or if we keep again in the hypotonic solution. It may again regain its shape, and then that process is known as deplasmolysis. So it was about the cell wall. In case of the bacteria, the cell wall is mainly made up of chitin, which is made up of two material, NAM and NAG, N-acetylmuramic acid and N-acetylglucamic acid, but glycosidic acid. But we'll discuss about this in later classes. So it was all about the cell membrane and cell wall. About cell membrane, one more very important thing is that about its structure is that the most accepted model of cell membrane was fluid mosaic model. Proposed by Singer and Nicolson. In 1972, in which they said that it is made up of the phospholipid and protein molecule, and the structure is something like this. That is, these dot-like or the circular structure of the phospholipid, and this structure is protein. So as these are the double layered, so it is a bilayer. More about this we will explore in our next class of 11th standard. So it was all about cell membrane and cell wall. Hopefully this class will help you to know some basics about the cell membrane and cell wall. Thank you very much.